Hey everyone, this is part B of the digestive system. So we're going to start talking about the throat in this portion and the esophagus and continuing on our adventure down the entire tube. So the pharynx is your throat, as you already know from respiratory. Food's going to pass from your mouth into the oropharynx and then into the laryngopharynx. Remember that the oropharynx and the laryngopharynx are a common passageway for both food and liquids and air, which kind of sets things up to be a little dangerous if something goes down the wrong tube. The pharynx is lined with stratified squamous epithelial. It has lots of mucus-producing cells in it. It has external muscle layers that are two different layers of skeletal muscle. The inner layer runs longitudinally, and then the outer layer is called the pharyngeal constrictors, or kind of rings that encircle the walls of the pharynx. The esophagus is a flat tube that is collapsed when it is not involved in the movement of food. So it is lying behind the trachea. It's kind of just hanging behind the trachea. It pierces the diaphragm at the esophageal hiatus. It joins the stomach at the cardial orifice, or where it dumps into the stomach. And that's where we find the gastroesophageal or cardiac sphincter that is allowing the food to pass into the stomach. That orifice is kept closed when you are not putting food into your stomach. If that orifice opens up, or that gastroesophageal doesn't keep it closed, that's what acid reflux is all about. Lots of mucus cells on both sides of the sphincter, the cardiac sphincter, to help protect the esophagus from the very acidic nature of the chyme in your stomach. Esophagus has all four alimentary canal layers. The mucosa in the esophagus is stratified squamous. It changes to simple columnar once it gets down to the stomach. The esophageal glands in the submucosa are what are secreting mucus that help to move the bolus of food. The muscularis externa, the skeletal muscle, is superior. It's mixed in the middle, and then it's smooth muscle on the inferior component of it. And rather than having a serosa as the outermost layer, it has a much tougher connective tissue called the adventitia. This is a cross section showing you those different layers of the alimentary canal. So you see the lumen in the very middle there. Then you have the mucosal layer, which is stratified squamous epithelial tissue, and that's the kind of dark purple layer. Then you have the submucosal layer, which is kind of like the lighter blue. Then you have the muscularis layers, the circular layer versus the longitudinal layer. Then you have the adventitia, which is the outermost layer of the esophagus. Just a different microscopic picture looking at a cross section showing you the junction between the esophagus and the stomach. So it's a little bit further on down there, and it's showing you how the the mucosal layer goes from being stratified squamous to being simple columnar. So heartburn. Heartburn is caused when the acid that's in your stomach bubbles back up into the esophagus, or is regurgitated into the esophagus. This is the first symptom of a condition called GERD, or gastroesophageal reflux disease. It can be caused by just eating too much or drinking too much. So if you just had like a really huge meal like Thanksgiving, something like that. It can also be caused by obesity. It can be caused during pregnancy. A lot of pregnant women say that they have problems with heartburn during pregnancy. It can also be an outcome of running. So when you're running and you're jostling your stomach around, that sphincter, that cardiac sphincter, opens up and allows things to bubble up. It can also be caused by hiatal hernia, which is an abnormality where the stomach actually comes up through that hiatal hiatus in the diaphragm. So there's an opening in the diaphragm that the esophagus passes through, and sometimes the stomach bulges backwards up into there. That can lead to esophagitis. It can lead to esophageal ulcers. It can even lead to esophageal cancer, specifically a type of cancer that's associated with heartburn is called Barrett's esophageal cancer. So what's going on as far as the digestive processes that are occurring in the mouth? So the throat and the esophagus are just tubes that are going to pass the food from the mouth to the esophagus or to the stomach. The major function of both of those organs is is swallowing. So we refer to swallowing as deglutition. It actually involves 22 different muscle groups, and there's two phases to this. So swallowing begins as a voluntary process that is initiated by your tongue. They call that the buccal phase because it's what happens in your mouth. And then it takes over once that bolus of food gets down to a certain portion of your pharynx. It becomes an involuntary process. And that's when it's called a pharyngeoesophageal phase. And this is controlled by the vagus nerve. So swallowing is the swallowing center is in the medulla and the lower pons. So they're showing you in this picture the whole process. So the bolus of food is green in here, and it's showing you how that food moves down, and that once you reach a certain point, it becomes an involuntary process, and peristalsis takes over, the rhythmic contraction and relaxation and contraction of relaxation of those muscles in the esophagus to move things down the throat. Swallowing is a really, really important issue. You cannot get nutrition in, and you can't even swallow your own spit if there's something wrong in that area. So even in the smallest facilities, they have ways of testing what they doing what they call a swallow test to make sure that they don't give a patient something to drink and they end up choking on it, or that they can get nutrition in. So there's just the different phases. So it's telling you about the buccal phase, where the upper esophageal sphincter is contracted. So we don't talk about that, but right there behind the epiglottis is the first of the series of sphincters. It's not on our models, and we never point it out in the lab, but there is an esophagus, a sphincter there that is closed. The tongue presses up against the hard palate, which forces the bolus of food down into the esophagus or to the oropharynx, and eventually down to the esophagus. So then that part right there is voluntary. Then you have the pharyngeal esophageal phase that begins when the tongue blocks the mouth, the soft palate and the uvula rise up so that food doesn't go back into your nasal passageways. The larynx rises up so the epiglottis flips over and blocks the trachea, and the upper esophageal sphincter relaxes, which allows the food to go down the esophagus. And then that phase continues. The constrictor muscles of the pharynx contract, and they force the food into the esophagus rather than going down into the trachea, and the upper esophageal sphincter is gonna open back up after the food is passed by. So now we're all the way down, and so we've been having this process of peristalsis, a rhythmic contraction that's moved that food all the way down to the stomach, and we finally get down to the stomach. The lower gastroesophageal sphincter, or the cardiac sphincter, opens up and allows the passageway of that food into your stomach. Let's take a look at the stomach for just a second, the gross anatomy of this. Remember that the stomach is nothing more than a temporary holding or storage tank. There's not a whole lot of absorption that occurs at the stomach, but there's a lot of digestion or breaking down that occurs at the stomach. So this is where we start the chemical breakdown of protein. So we started the chemical breakdown of carbohydrate in the mouth. Now we're going to start the chemical breakdown of protein in the stomach. So the stomach is going to convert your food into this substance that's called chyme. It's basically like a milkshake. It's going to churn it all up. Your empty stomach has a volume of about 50 milliliters, but it can expand to 4 liters. What allows it to do that are the expansion folds called the rugae. Major regions of the stomach include the cardiac region, which is right there at the opening of the esophagus into the stomach, the fundus, which is the dome-shaped area, the body, which is the majority of the stomach, and then the pyloric part, which is the part that starts to go right into the duodenum. And so in lab, we just call that the pyloric region, but it is actually made of the antrum, which narrows into a canal and terminates in the pylorus. And then right there at that termination point is where you find the pyloric sphincter, which is controlling the movement of food from your stomach into your small intestine. So showing you all those different areas of the stomach. And then looking at an actual stomach in a cadaver. You also notice the greater curvature and the lesser curvature. The greater curvature is on the bottom side of the stomach. The lesser is up on the top. The mesenteries extend from those curvatures and help to anchor the stomach to the other digestive organs. The lesser omentum is the sheet of fat that runs from the lesser curvature down to the liver that also helps to anchor it. The greater omentum is a true apron of fat that sits over top of the intestines and the spleen and the transverse colon. It drapes from that greater curvature. It blends with the mesocolon, the mesentery that anchors the large intestine to the abdominal wall, and so the greater omentum has a lot of fat and lymph nodes in it. The autonomic nervous system is supplying innervation to the stomach. The sympathetic fibers from the thoracic splanchnic nerves are relayed through the celiac plexus, and the parasympathetic fibers are supplied by the vagus nerve. So remembering from 210, in the autonomic nervous system, the sympathetic typically speeds most systems up, but the two systems it actually slows down, or three systems that it actually slows down, are digestive, urinary, and reproductive. Typically, the parasympathetic slows systems down with the exception of the digestive, urinary, and reproductive. So those three systems are flipped in the commands of the sympathetic and parasympathetic fibers. The blood supply, the celiac trunk coming off of the abdominal aorta is the portion that's supplying blood. The gastric and splenic branches are supplying blood to the stomach, and then we had the veins of the hepatic portal system, one of which was coming off of the stomach, taking that low oxygen but high nutrient rich blood over to the liver for offloading. The stomach wall has the regular four tunics, however, the muscularis and mucosis are modified. The muscularis externa has the typical circular and longitudinal layers, but it also has an additional third layer that is oblique. So it really allows the stomach to turn into a blender and increases the physical breakdown of the food. The mucosal layer is also modified. It has a simple columnar epithelium that is entirely composed of mucus cells, and it secretes two-layer coat of alkaline mucus. So the stomach, to be able to digest the protein or break the protein down, the pH in the stomach has to go really, really low. And if that happens and the stomach itself is exposed to that low pH, it will also break the stomach down because the stomach is made of protein. So obviously we have to have something that's protecting the stomach from that happening, and that's what this alkaline mucus does. It's dotted with gastric pits that lead into the gastric glands. The gastric glands are what are producing the gastric juice. So there is a picture of one of these little layers, so you can kind of get the idea of all the different layers with the surface epithelium and the submucosa, the muscularis externa with that extra oblique layer on the inside, and then finally the serosa on the outside. The types of glands that you have, so there's glands that are in the fundus and the body region that make most of the gastric juice. Those glands include secretory cells called the mucous neck cells, the parietal cells, the chief cells, and the enteroendocrine cells. So here is one of those gastric pits that we're talking about, and then you can see how it goes into the gastric gland. So if you look over on the left-hand side of the picture, it's got the brackets. So the indention that leads down to it is called the gastric pit from the surface, and then that area that's below that's called the gastric gland, and it's made of 4 different kinds of cells. One of those cells is called the mucous neck cell. It secretes a thin acidic mucus that they're not real sure what it does. This is unlike the alkaline mucus that is being secreted that's coating the inside of your stomach. Then you have the parietal cells. The parietal cells are going to secrete hydrochloric acid. Hydrochloric acid has a pH between 1.5 and 3.5, and what it does is it denatures the protein. So it takes it and it uncoils the protein so that it can be further cleaved apart. It's going to activate pepsin. It's going to break down the cell wall of any plant material that you've consumed, and it helps to kill any bacteria. Most bacteria can't survive at that low of a pH. You also have secretions from the parietal cells called intrinsic factor. This is a glycoprotein that is entirely necessary for you to be able to absorb B12 from your small intestine. Chief cells. Chief cells are going to have a couple of secretions. One of them is called pepsinogen. So remember, ogen always means the inactive form. This is the inactive enzyme that activates pepsin. That's activated to pepsin by hydrochloric acid. So hydrochloric acid is going to convert pepsinogen into pepsin. And then pepsin also has the ability through a positive feedback mechanism to convert itself into pepsinogen into more pepsin as well. You also have some lipases that are secreted. These are going to be enzymes that help to digest lipids. Then you have the enteroendocrine cells. These are secreting chemical messengers into the lamina propria. They act as paracrines. So two of those include serotonin and histamine. The hormones that are secreted by the enteroendocrine glands include somatostatin, which also acts as a paracrine or has an effect in the local area. That's what paracrine means, as well as gas. So taking a close-up look at the gastric gland here, so you get the idea of all the different color cells. So they're showing you a parietal cell, which is blue, got lots of mitochondria in it, and they're showing you the chief cells, which are purple, the enteroendocrine cells, which are way down there at the bottom, they're green. So they're giving you all of those different kinds of cells. So remember that the parietal cell is going to release hydrochloric acid, and the hydrochloric acid is going to convert the pepsinogen into the pepsin. So your stomach, as I mentioned, has a mucosal barrier. We have to make sure that the stomach is protected by the low pH of the stomach that's created by the hydrochloric acid. That makes a couple of different ways that it's protecting the stomach. Number one, it creates this thick layer of bicarbonate-rich mucus. It also, another way that we're protecting that stomach is with the tight junctions that are between the epithelial cells so that none of the very acidic gastric juices can leak in between and get to those underlying tissue. Damaged epithelial cells are very quickly replaced by the stem cells. The stomach lining replaces itself every 3 to 6 days, so any damaged cells would be leaky cells, and those would be a liability, so they are replaced very quickly. Gastritis is inflammation that's caused by anything that breaches the stomach's mucosal barrier. So an example of that is when you've got maybe like the stomach flu or something like that, and you've lost a lot of that mucus that's in your stomach when you've been vomiting a lot. Peptic or gastric ulcers, these actually erode through the stomach wall, and if it perforates the wall, it can lead to peritonitis and a hemorrhage. Most ulcers, which is surprising to most people, and most doctors still don't know this, is that ulcers are caused by a bacteria. That bacteria is called Helicobacter pylori, and it is picked up easily in the environment. Lots of people have it. It's long as you have an intact lining to your stomach, it isn't a problem. But if there is a little crack in that lining or there's a little eroding away in that lining for whatever reason, then that's going to allow that bacteria to get in and cause an ulceration. One of the things that can cause that lining to be broken is aspirin. Aspirin tears up the lining of the inside of your stomach. So a lot of people choose to take enteric or coated aspirin so that it doesn't have that effect on their stomach. There's a picture of an ulcer. And there's the bacteria that's responsible, H. pylori, that's responsible for causing ulcers. So there's a guy that won the Nobel Prize for this back in the 1990s. He actually gave himself an ulcer by ingesting H. pylori so that he would be able to demonstrate it was the causative agent of ulcer. Up to that point, they thought ulcers were caused by stress. So if your doctor tells you that it's your stress that's causing your ulcer, tell him that you need an antibiotic. All right, so the processes that are carried out by the stomach. Number one, it carries out the breakdown of food, and it serves as a holding facility for that food. It's going to slowly deliver that chyme to the small intestine. It's going to denature the protein. So it starts the digestion of protein by uncoiling the protein, and that's done by hydrochloric acid. Pepsin is going to carry out the enzymatic digestion of proteins. For example, milk protein or casein is broken down by a different enzyme, one called renin in infants. So renin is not the same renin that we saw with the renin-angiotensin system. Notice that it has two N's, so it's a different renin. If you've ever had a little one spit up on you after they have eaten, you notice that it's real curdy, it's real chunky, and that's what causes the renin. That's what causes that is the renin. One of the other things that your stomach does is it allows the absorption of alcohol and aspirin, and it goes right into the portion of the hepatic portal vein that's coming off the stomach, going right to the liver, which is why when people abuse liver or abuse alcohol or aspirin over a long time, their liver is the first thing that starts to suffer from that. The only stomach function that's essential to life is the secretion of intrinsic factor, which I mentioned before, which is necessary for vitamin B12 absorption. Why do you need B12? You need it for the maturation of your red blood cells. When you don't have enough intrinsic factor, it can lead to a condition that we talked about in the blood chapter called pernicious anemia, and we treat this with B12 injections, or there's a nasal spray of it now. There's pills and such as well. So how do we regulate the amount of fluid or secretions that are happening at the stomach? So we talked about the salivary glands in the last lecture, and remember, you make approximately 1.5 liters or 1.5 liters of saliva every day. You make a little bit more than 3 liters of gastric juice every day. So I want you to kind of in your mind keep track of how much we're adding to this tube. So far, just from the salivary glands and the stomach, we've added 4.5 liters of fluid. So what is this regulated by? What's the secretion of gastric fluids regulated by? It's regulated by neural mechanisms. So the vagus nerve, which is typically stimulated, it's typically inhibitory, and when it's stimulated, it causes the secretion of gastric solutions, and then sympathetic stimulation decreases them. So if you've ever been in fight or flight, you notice that your mouth gets really dry, and you notice that you're not hungry. And that is part of making you prepared to flee for your life or stand and fight for your life. The hormonal component of this, gastrin, is what stimulates enzyme and hydrochloric acid secretion, and then gastrin antagonists are secreted by the small intestine, so that once the food has left the stomach, we stop the secretion of solutions in our stomach because we don't need it anymore. Gastric secretion is broken into 3 phases. There's the cephalic phase, which is a reflex, which is what Pavlov did all his work with the dogs. There's the gastric phase, and there's the intestinal phase. So the cephalic phase is a conditioned reflex that's triggered by the smell, the taste, the sight, or the thought of food. So if you know anything about Pavlov and his dogs, he was able to take a meat powder, and every time he gave the dogs the meat powder, he rang a bell. And so pretty soon all he had to do was ring the bell, and the dogs would start salivating like they were going to get the meat powder. So they learned to associate the bell ringing. You've seen this as well at home if you have a cat. If you have a cat and you use a can opener to open a can of cat food one time, that cat will remember that for all of eternity. And every time you use a can opener, the electric can opener, the cat will come running, right? So that's that idea too. And you and I do this as well. So there's certain things when we smell them or we see them or we even think about them, they make our stomachs start to secrete solutions. The gastric phase lasts for about 3 to 4 hours, and this is where the majority of the gastric juices are released, of course, because you have stuff in your stomach. The stimulation of the gastric phase is caused by the distension of stretch receptors, both long and short reflexes that happen at the stomach. It's also caused by the chemical presence of partially digested proteins, caffeine, low acidity, any of those kinds of things are going to cause the stomach to start secreting gastrin, which is part of the gastric juices. The release of gastrin is going to initiate the hydrochloric release from the parietal cells, and that activates the enzyme secretion. So this is going to encourage the parietal cells to secrete hydrochloric acid by binding to the receptors on the parietal cells and stimulating the enteroendocrine cells to release histamine. There's a buffering action of ingested proteins that causes the pH to rise, which activates even more gastrin secretion. How do we inhibit the gastric phase? Well, low pH inhibits the gastric phase. This is what occurs between meals. So this also occurs during digestion as a negative feedback mechanism. So the more protein that you consume, the more hydrochloric acid that's secreted, which causes a decline in the pH, which inhibits gastrin secretion. The intestinal phase is what happens after the food leaves the stomach and starts to head into the intestine. So as that food is moving into the stomach, it begins with a brief stimulatory component, which is followed by inhibition. So stimulation of the intestinal phase begins with partially digested food entering into your duodenum and causing a brief release of enteric or intestinal gastrin. And this encourages the gastric glands of the stomach to continue their secretory events. The stimulatory effect is very brief and is typically overridden by the inhibitory stimuli as the intestine starts to fill up. So essentially what it's saying, and what they haven't gotten to yet with all of that, why do we want that to happen? Because we're slowly emptying the stomach, right? And so if the first little bit of food that went from your stomach into your intestine caused your stomach to stop releasing gastrin, then you wouldn't finish digesting what's in your stomach. Right, so we want it to continue while we start the initial part of moving that stuff over, but there is a point where we don't need for that to happen anymore. So the inhibition of the intestinal phase, there's four major factors in the duodenum that cause the inhibition of gastric secretions. When the duodenum becomes distended because of the entry to chyme or the milkshake that you made out of your food, the presence of acidic chyme, the presence of fatty chyme, the presence of hypertonic chyme, all of those things are going to start causing the stomach to stop making its secretions, meaning that we are now moving on into the intestines. The inhibitory effects protect the intestine from becoming overwhelmed because we don't want too much chyme to be entering at once. The inhibition is achieved two ways, the enterogastric reflex and the enterogastrone. So the enterogastric reflex, when the duodenum inhibits acid secretion in the stomach by using its own internal nervous system, the short reflexes of the enteric nervous system, and the sympathetic nervous system and vagus nerve long reflexes. Enterogastins are released by the duodenal enteroendocrine cells. There are two of these that are important to know about. Then they are going to inhibit gastric secretions. One is secretin and the other is cholecystokinin or CCK. So taking a look at this is the neural and hormonal mechanisms that are regulating gastric juice release. And so it's showing you all of the different stimulatory effects for the cephalic, the gastric, intestinal phases, and then showing you what causes those to become inhibited. Here are the list of hormones and paracrines that you're responsible for. So cholecystokinin and so on, GIP, gastrin, histamine. Some of these you may already have on your chart. Some of them you may not. So you need to make sure that you add these and you are responsible for knowing where they are produced, what's the stimulus for that, what's the target, and what do they do. So 2 pages of that. So make sure you read through those and you understand those different hormones. How do we form the hydrochloric acid in the stomach? So parietal cells are going to pump hydrogen ion from the carbonic acid breakdown into the stomach lumen via the hydrogen potassium ATPase pumps, better known as proton pumps. That's why all of those drugs like Nexium and all of those different drugs that are out there that are acid reducers, those are called proton pump inhibitors. All right, so you see the tie-in there. So that's how we're making this hydrochloric acid, is using these proton pumps. So as the hydrogen ion is pumped into the stomach lumen, the bicarbonate is exported back to the blood via the chloride ion and the bicarbonate antiporter. This results in an increase in the bicarbonate in the blood leaving the stomach, and we refer to that as the alkaline tide. The chloride ion is pumped out of the lumen to join the hydrogen ion to form hydrochloric acid. So they're showing you how that little process happens in the gastric gland. What's the response to the stomach to filling? So as your stomach fills up, it stretches, right? And you have stretch receptors that are detecting that. There's 2 factors that cause pressure to remain constant in your stomach until you hit about 1.5 liters of food. One is the receptive relaxation. That's the reflex-mediated relaxation of the smooth muscle that's coordinated by the swallowing center of the brainstem. And the other is gastric accommodation, which basically means that the smooth muscle in your stomach can continue to stretch and stretch and stretch without increasing the tension or contractions. All right, so what is moving things through your stomach? That peristalsis that started up in the pharynx continues with the stomach. So you've got those wave-like motions going through the stomach, and it's pushing the food towards, or what's left of the food now, the chyme, towards the pyloric end. You've got about 3 waves of peristalsis happening per minute. Your intestines and your digestive system has its own set of pacemaker cells, just like we saw with the heart. These are called the enteric pacemaker cells. The old name for this were the interstitial cells of Cajal. They are linked by gap junctions, and they are controlling the entire muscularis layer. So again, not your general nervous system, your central nervous system, but its own special set of neurons that are firing and causing the waves to happen, because the waves are muscular contractions. And so you also have distension and gastrin that increase the force of contraction. The contractions are most vigorous and powerful near the end of the stomach at the pyloric area. 30 milliliters of chyme is produced at a time, and it's delivered into the duodenum in about a 3 milliliter little bolus. So it's about a tablespoon, more or less. The other 27 milliliters is forced back into the stomach. So we don't want this chyme to enter in to the duodenum too fast. The main function of your small intestine is absorption. So we want to make sure that we don't overload that small intestine. So we want just a little bit to go in at a time so it has an opportunity to be fully absorbed. So the only thing that's going to pass through that pyloric sphincter or pyloric valve are liquids and small particles. So big chunks of things don't go through. So here they're showing you propulsion that's happening with the pyloric valve that's closed. The stomach is twisting and grinding. Very vigorous peristalsis that happens in the stomach. The pyloric end of the stomach acts as a pump that delivers that little bit of chyme through the pyloric sphincter into the duodenum, and then it shuts and everything's pushed back up. So we're only letting a little bit go through at a time. The duodenum can prevent overfilling by controlling how much chyme enters it. So the duodenum is not a very long structure. It's about 10 inches long. It's about the thickness of a garden hose. So we don't want a whole bunch to dump into it all at once. So the duodenum controls how much is going in because it has stretch receptors and chemical signals. The enterogastric reflex and the enterogastrones inhibit the gastric secretion and the duodenal filling. So your stomach takes about 4 hours to empty. If there's fat and protein in your food, it could take up to 6 hours. If all you have in your food is carbohydrate, it moves through much, much faster. So this is looking at the neuronal and hormonal factors that inhibit the gastric emptying. So we don't want it to empty too fast. So vomiting or emesis is caused by extreme stretching of the stomach and intestinal irritants like toxins from bacteria, too much alcohol, spicy food. Some drugs make you sick to your stomach. Chemicals and sensory impulse stimulate the emetic center in your medulla. So vomiting is actually a reflex. You vomit because your brain says something's not right and you need to get it out. Excessive vomiting can lead to dehydration and an imbalance in your electrolytes and your acid bases. That's called alkalosis when that happens. Moving right along, going to the liver, the gallbladder, and the pancreas. These are all accessory organs that are associated with your small intestine. Food does not move through them. Food moves by them. The liver has a lot of different functions. The main function that we're going to talk about right now has to do with the production of bile. And what bile is an emulsifier. Your gallbladder is where we store the bile. So the liver makes the bile. The gallbladder stores it. The pancreas also supplies enzymes that we need to digest the chyme even further as it enters into the small intestine. It also has a neutralizer in the form of bicarbonate. So let's talk about your liver in particular. Gross anatomy of it, largest gland that you have in the body. It weighs about 3 pounds. It has 4 primary lobes, the right, left, the caudate, and the quadrate. The falciform ligament is like a white ligament that separates the right lobe from the left lobe. The right lobe is the largest lobe, and it also suspends the liver from the diaphragm, kind of like a chandelier, and it attaches it on the bottom side to the abdominal wall, so it's not just swinging around in there free. The round ligament is the remnant of your fetal umbilical vein, as well as what's left of the falciform ligament when it comes off of the edge of the liver. So that's what they're showing you. Want to direct your attention to the bottom right-hand side where they're pointing out the round ligament. So while it's in the liver, it's called the falciform ligament. When it comes off of the liver, then it's referred to as the round ligament. Here we are looking at the back, so you can see the caudate lobe up at the top, the one that starts with a C, and the quadrate lobe down beside the gallbladder between the round ligament or falciform ligament and the gallbladder is where you have the quadrate with a Q. The lesser omenum is what anchors the liver to the stomach. You have a hepatic artery and vein that enter the liver at the porta hepatis. You also have bile ducts. So bile ducts are what are delivering the bile ultimately to the duodenum to emulsify or help break down the fat. The common hepatic duct is what carries the bile from the liver to the cystic duct, which carries it to the gallbladder, and then the bile duct or the common bile duct is what comes off of the cystic duct and goes down to the duodenum. So that's what they're showing you here. The common hepatic duct is just that little green chunk right there below that blue vessel, and then you can see the cystic duct, which is the neck of the gallbladder. And if you can imagine where that common hepatic is cut off, if you could imagine that continuing on down, that would be what goes down to your small intestine, to the duodenum. When we look at the liver microscopically, it's made of little lobules. These are hexagonal structures, and they are also considered the functional unit of the liver. Those are made of liver cells that are called hepatocytes. They're kind of like plates. The cells are kind of flat. They filter and process the nutrient-rich blood that's brought in from the hepatic portal system. There is a central vein that's located down the center of it. A portal triad is on each corner of that lobule, and it consists of a hepatic artery, which is bringing oxygen to the liver tissue itself, a branch of the hepatic portal vein, which is bringing the nutrient-rich blood in from the intestines, and a bile duct, which is receiving bile from the liver, specifically through some structures, some little passageways that it's delivered down to the cystic duct or to the common hepatic duct, then to the cystic duct. Those little passageways are called the bile caniculi. So there's what it looks like underneath the microscope. Close up of that so you can see the central vein. The liver has a lot of leaky capillaries in it. So the capillaries are what they call sinusoidal. If you remember that from our capillary discussion earlier this semester, they're located between the hepatic plates. So as blood comes in from both the hepatic portal vein and the hepatic artery, it sort of percolates through these areas and empties into that central vein. You also have some special fixed hepatic macrophages in the liver that are called stellate macrophages. And they are serving to remove debris and old red blood cells. There is the picture that we looked at in our lab as well. This is the microscopic anatomy of the liver. So when you look at the corners, you see a red tube, a blue tube, and a green tube, and that collectively is called the portal triad. The hepatocytes have increased amounts of rough and smooth endoplasmic reticulum. They have increased amounts of Golgi, peroxisomes, and mitochondria. So what are some of the other things that liver cells do besides make bile? Well, they do make about 900 milliliters of bile a day. So add that to our 1.5 liters of spit and our 3 liters of gastric juice. So we were at 4.5. Now add another liter more or less. So we're at 5.5 liters of fluid here. The liver processes bloodborne nutrients. It takes the glucose and stores it as glycogen. It also helps to make plasma proteins like albumin. It stores our fat-soluble vitamins A, D, E, and K, and it is known to be the detoxifier. So it converts things like ammonia into urea. It is also where it detoxifies drug and alcohol. What is bile? Bile is a yellow-green alkaline solution that is made of bile salts, and bile salts are a derivative of our cholesterol that we consume and that we make. So this is why we have to consume a certain amount of cholesterol every day, and we have to make a certain amount of cholesterol every day. So this is what is going to allow us to emulsify fats. So here's an important thing to think about. Mostly what we have been dumping into the digestive tube along with your food is a lot of water or a lot of water-based products. And you guys know that fat and water don't mix. So to allow for the fat and the water to mix, bile salts allow that to happen. It's basically a detergent that breaks the big globules of fat down into little bitty, teeny tiny things called micelles. And the micelles are small enough to pass over the digestive lining, go into the lacteals, and be carried into the lymph. So that's what bile salt is. That's one component of the bile. The other one is bilirubin, which is the pigment that is formed from the breakdown of hemoglobin molecule when red blood cells are broken down. So remember that they go to the spleen, and the spleen kind of filters out any of the red blood cells that are looking a little rough and aren't able to do their job anymore, and we recycle most of the components of that, and one of them is heme, and bilirubin is the pigment that's formed from that heme. Bacteria break down in the intestine. They form a stercobilin that gives your color to your waste material. So the waste material that you and I produce should be a brown color. If it's not a brown color, it indicates that the liver's not working correctly. Some other things that are in bile is cholesterol, triglycerides, phospholipids, and electrolytes. So we recycle a lot of this bile. It's what's called the enterohepatic circulation. This is the mechanism that we use to conserve our bile salts. So bile salts are reabsorbed into the blood by the ileum, which is the last part of the small intestine. So we add the bile up in the duodenum, and then we reabsorb the bile salt portion at the very end of it once we've emulsified the fats and they've been used or they've been absorbed. We return that to the liver via the hepatic portal blood, and then it's resecreted as newly formed bile. We're able to recycle about 95% of our bile salts, so it's only about 5% that has to be synthesized. So some homeostatic imbalances of the liver, hepatitis. These are typically viral. They can be caused by drug toxicity. They can also be caused by wild mushroom poisoning. So hepatitis, there are, I don't remember how many versions now. They're up to like hepatitis F and G and other countries. You and I are familiar with A, B, and C, which is what we typically see in the United States, North America. Cirrhosis is a progressive chronic inflammation from chronic hepatitis or alcoholism. The liver becomes fatty and stringy or fibrous, and it causes a disease called portal hypertension. The liver transplants are pretty successful, but livers are relatively scarce. Most people are using theirs. The gallbladder. The gallbladder is a thin-walled muscular sac that's on the bottom side of the liver. Its function is to store and concentrate the bile that was made by the liver. It does that concentrating part by absorbing some of the water that's in the bile that's produced by the liver and the ions as well. So it has lots of honeycomb folds in it. So it's very similar to rugae that allow it to expand. Muscular contractions are what are going to release the bile via the cystic duct, which is going to go down to the common bile duct into the duodenum. So we only want bile to be released when there's fat in the chyme. We don't want it to be released any other time because it is an emulsifier, and so bile that's being released when you don't need bile often leads to intestinal distress. Gallstones. These are caused by too much cholesterol or too few bile salts, and they can obstruct the flow of bile from the liver. They're really painful because a lot of times they have little sharp sticks or crystals to them, and so when the gallbladder contracts, it gets poked from the inside. Obstructive jaundice is when you have a blockage that is causing the bile salts and the pigments to build up in the blood, and it causes the skin to yellow. So jaundicing can be a sign that the liver's not working, but if the liver's working fine and the patient is getting jaundiced, it's because they have a stone stuck somewhere. Typically, they know that because it's very painful. The treatment for gallstones are different types of dissolving drugs. Um, Lithotripsy, like they do with the kidneys, they can try that, some ultrasound vibrations, laser vaporization, but typically what they do is they're just going to do surgery. What about your pancreas? So your pancreas is located retroperitoneal, so it is outside of that digestive sac. It's deep to the greater curvature of the stomach. The head is encircled by the duodenum. The tail butts up against the spleen. The exocrine function of the pancreas is to produce pancreatic solution or juice. It does this from some cells that are called acini cells that produce zymogen granules that contain the proenzymes needed to digest all of the different macromolecules. And then there's a duct to that that's going to secrete the solution into the duodenum via the main pancreatic duct, the thing that looks like a fish bone, the backbone of a fish running through the middle of it, and then into smaller ducts that produce duct cells that produce water and bicarbonate. So that's the exocrine function. And remember that the definition of an exocrine gland is that it makes some product and it secretes it through a duct. So that's what makes it exocrine. But the pancreas is unique in that it also has endocrine function. So we already learned about the endocrine function when we talked about y'all's favorite topic, which was hormones, where it secretes insulin and glucagon by the pancreatic islet cells. So the pancreas is unique in that. This is just a slide showing you the acinus, which is that round structure, the cells that are making the zymogen granules that are being secreted into the duct, where it's going to mix with the bicarbonate and form pancreatic solution or juice. There it is in an electron or in a micrograph. So what is the composition of pancreatic juice? We make between 1,200 and 1,500 milliliters a day. So add 1.5 liters to the amount of fluid that we have now dumped into our intestinal tube. It is a watery alkaline solution with a pH of about 8, and its job is to neutralize that very, very acidic chyme. So we brought the chyme down to a pH of about 1 so that we could start the denaturing and breakdown of proteins. And now that acidic chyme is being put into the small intestine, and we're going to have to bring that pH back up because the small intestine, the tissue lining the small intestine is not able to handle that level of pH. It's got to be brought back up to a neutral level. Some other things you find in pancreatic solution are electrolytes, primarily bicarbonate, as well as the digestive enzymes. So here's the big deal about the pancreas. It releases all of the digestive enzymes that we need to break down all of our macromolecules. So we may have some preliminary breaking down of carbohydrate in the mouth with salivary amylase, but pancreatic amylase is going to finish that up. We had some preliminary breakdown of proteins in the stomach where we denatured them or uncoiled them and chopped them into big chunks, but proteases are going to break those down into little teeny tiny pieces, or the precursors are called proteases for proteins. They have to be secreted in the inactive form because they are breaking down protein. And again, your pancreas is made of protein. Lipases, which are going to allow us to break down the lipids after the bile has emulsified it, and nucleases, which are going to allow us to break down the nucleic acids. Now, we don't get any caloric value off of nucleic acids, but we do use the spare parts to make new DNA. So we need to be able to break down those 4 macromolecules, and the big player in doing that is the pancreas. So you see why the pancreas and having pancreatic problems is a major, major issue. Proteases, again, are secreted only in the inactive form. They are activated after they reach the duodenum. Enteropeptidase is the enzyme that's bound to the plasma membranes of the duodenal epithelial cells, and it activates the pancreatic protease trypsinogen to trypsin. So again, OGN always tells us in the inactive form. Once trypsin is activated, it then activates more trypsinogen, but it also activates procarboxylpeptidase to activate the carboxypeptidase, and chymotrypsogen to activate chymotrypsin. So there you have it. We're looking at the inside of the small intestine at the duodenum. You see the little opening where the pancreatic solution is injected into the small intestine. It's showing you these different inactive forms that are found in the small intestine. They're released by special cells in the brush border of the small intestine called enteroendocrine cells. They release those in the inactive form, and then we start activating those so we can break down protein. So how does that bile and pancreatic solution get into the small intestine? The two of them, the bile duct and the pancreatic duct, are going to unite in the wall of the duodenum. Then they're going to fuse together in a structure called the hepatopancreatic ampule. The ampule actually opens up in the duodenum through a papilla. It's called the major duodenal papilla, and there is a sphincter that controls the entry of the bile and the pancreatic juice called the hepatopancreatic sphincter. That used to be called the sphincter of Oddi. The accessory pancreatic duct we looked at in the lab. Remember, that's kind of like the side street that comes off the main pancreatic duct. Not everyone has that, but it also dumps directly into the duodenum. So showing you that main pancreatic duct going right down the very middle of the pancreas, and then you see the accessory. I think it's estimated 3 to 8% of the population has that particular duct, but you can see that duodenal papilla right there. And of course, the sphincter is what's opening and closing and controlling the movement of that fluid into the small intestine when necessary. How do we regulate the bile and the pancreatic secretions? They're both regulated by neural and hormonal controls. So the hormonal controls include cholecystokinin or CCK and secretin. Bile secretion is increased when you have a large amount of bile being returned by enterohepatic circulation. Remember, we're recycling that. When there is secretin from the intestinal cells that have been exposed to hydrochloric acid and fatty chyme, that stimulates the gallbladder to release bile. So whenever we have fatty material in the duodenum, that is a signal for the gallbladder to contract. The hepatopancreatic sphincter is closed unless digestion is active. So we don't want bile or pancreatic solution dripping into an empty duodenum. The bile, again, is stored in the gallbladder and is released to the small intestine only through contraction. So this last little series of slides is showing you the mechanisms that are promoting the secretion and the release of bile and pancreatic juices. So it's all coordinated. We don't want bile or pancreatic juices put into the intestine unless we actually need them. So there's a step-by-step for CCK and for secretin, and they're two different colors. So you can read through all of that and try to make some sense of it.
Okay, I'll see you all for part C. Bye.